Babies are born in hospitals and homes every day. Each bears a genetic heritage. Each possesses traits of parents and grandparents and great-grandparents and countless ancestors. And yet each child is unique. Each is a combination of traits. Never before have such combinations existed. Never before have the genetic constellations that define their bodies found expression. Based simply on physical appearance, about the only way to tell an infant boy from an infant girl is to look at their genitals. Along with the other parts of their reproductive systems, their genitals are not essential to the survival of their bodies. They are like dormant buds that ultimately will allow these children to reproduce. They exist to complement each other. Young children grow and develop at a rate that, at least to their parents, is amazingly fast. They quickly become aware of differences between boys and girls. Both pass through a phase when they are not at all interested in the opposite sex. As growth and development inevitably continue, and the years pass, these feelings will also pass. Profound changes begin. The children take on forms that are much less like each other. This time of life is called puberty. No one knows exactly how puberty starts, but we do know that the driving forces of the change are chemicals called hormones, and that the changes begin in the brain. Under the brain's influence, the pituitary gland begins to produce hormones that enter the bloodstream. Eventually, the hormones will reach the primary sex organs. In a male, the primary sex organs are the testes, suspended outside his body in a sac called the scrotum. In a female, the primary sex organs are the ovaries, located inside her body. The ovaries and testes react to the pituitary hormones by producing their own hormones, which travel throughout the bodies of the children. In males, hormones produced by the testes will cause the voice to deepen. They will cause hair to grow where it hardly grew before. They will cause certain of his muscles to develop, giving him masculine contours. They will affect him in a variety of rather unimportant cosmetic ways, but most important, they will cause his genitals to grow and develop. When boys are going through the changes in puberty, there is sometimes a release of fluid called semen during the night. This is commonly called a nocturnal emission or a wet dream. This is a natural occurrence and only means that more semen has been created than can be stored or reabsorbed by the body. A mature male has two fundamental roles in the reproductive process: to produce sperm and to deliver these sex cells into the female's reproductive tract. Sperm cells are created by the testes, which hang outside the body because sperm creation depends on the organs being slightly cooler than the rest of the body. At first, the cells from the testes contain the same amount of genetic material found in other body cells. Through a series of cell divisions called meiosis, that amount is cut in half. Finally, the cells become sperm. A typical sperm cell has a head, which contains the genetic material, a body to provide energy, and a tail, or flagella, to propel the cell. Sperm are produced at an amazing rate, about 200 million cells every day in an average adult. The sperm will be carried by a system of tubes that leads up into the body. Several other glands that produce the rest of the semen connect to this system. Also joining is the urethra, a tube that comes from the bladder. Finally, the tube exits the body's interior, surrounded by the penis. The penis is made of soft, sensitive tissues. It serves as a conduit for urine and for the delivery of sperm when maturity is reached. The penises of some men look like this, and some look like this. At birth, boys have a fold of skin, called the foreskin, surrounding the tip of the penis. Many doctors believe that the foreskin makes the penis more susceptible to infection and remove it at birth. This is called circumcision. If left on, it does not interfere with the male's ability to deliver his sperm into the female's reproductive system. To deliver sperm, the penis must temporarily change. Inside it are chambers of spongy tissue. When the male is sexually aroused, blood vessels that supply the chambers open wider than usual. The tissue fills with blood. At the same time, some of the veins that normally drain the chambers are partially closed, and the organ becomes stiff and erect. The male cannot urinate when his penis is erect. The tube that leads from the bladder is shut off so that no urine can pass. In this rigid state, the penis is ideally suited to deposit sperm into the female's reproductive system. When the male is no longer aroused, his penis returns to its usual state. When a girl starts going through puberty, there is a very marked happening, the beginning of her menstrual cycle. A girl usually has her first period sometime between 10 and 14 years of age, sometimes later. For many girls, their first period is an important signal that they are really growing up. In fact, it is one of many steps to adulthood. The hormones that the female's ovaries begin to produce at puberty will cause her breasts to develop. The hormones will cause hair to grow where it hardly grew before. They will change the shape of her body to give her feminine contours and a structure adapted to the delivery of children. And they will cause her to begin menstruating. What exactly is going on during the menstrual period? Why does it happen? And what is it for? To answer these questions, let's examine the tasks of the female in reproduction. She has three major roles. One is to produce ova, the female sex cells. This task is performed by the ovaries. Another task is to receive the male's sperm. This is done by the vagina. The third task is to provide an environment for growing a new human. This environment is the womb, or uterus. The menstrual blood comes from the uterus. Its appearance is the most obvious part of a complex cycle of events that is essential to reproduction. Because the cycles of a young woman are very often irregular, we will describe a typical cycle of an adult. The menstrual cycle is controlled by hormones and has two important aspects. One is the development of a mature sex cell, an ovum. Unlike the male, the female already has all her sex cells at birth, although they are immature and rest dormant in her ovaries. Both ovaries are filled with hundreds of thousands of follicles, each containing an immature egg cell. First, the follicle surrounding an ovum grows and swells. It migrates to the edge of the ovary, forming a small bump on the surface of the organ. It begins to undergo meiosis, the series of divisions that will reduce its genetic material by half. Fluid rapidly accumulates in the follicle, and its walls become thin. About 2 weeks into the cycle, the follicle breaks, releasing the ovum. This release is called ovulation. The ovum is transported down a passage called the fallopian tube, which leads to the uterus. Meanwhile, under the influence of hormones, a fine network of glands and blood vessels has developed on the wall of the uterus. If fertilization takes place, the egg will come to rest here. But usually, no fertilization occurs. In that case, this newly created network breaks down and is discharged to the outside as the menstrual flow. Watch the calendar on the side as we go through the cycle again. Day 1 is the first day of bleeding. This menstruation lasts 4 to 5 days. Day 6 through 13 shows the follicle growing and swelling in the ovary until around day 14, when it breaks. Within a few days, if the egg is not fertilized, it dies in the fallopian tubes. Any remnants will be washed down into the uterus, and after approximately 28 days, the cycle starts again. The menstrual discharge flows out of the body through the vagina. The vagina is a muscular canal that leads from the uterus down to the external genitalia. It receives the male's penis and sperm. Some women have a fold of thin tissue called the hymen that covers all or part of the opening to the vagina. During rigorous physical activity or at first intercourse, it may break and bleed slightly. In front of the vagina is the urethra, leading from the bladder to the outside and through which urine passes. In front of this is a small, very sensitive bulb of tissue called the clitoris. Surrounding all these are 2 pairs of skin folds called the labia. Although a young girl's body may now have the capacity to become pregnant, it is really not ready to bear children. A very high proportion of prematurely born babies come from teenage mothers. A number of years must pass. Years of physical development, years of mental development, before people are completely mature. As this happens, they become more and more interested in the opposite sex. When people are mature enough to raise children successfully, they may choose not to. There are several ways of preventing pregnancy, but eventually, the couple may decide that it is time to have a child. Sexual arousal is an intensely personal, complex process involving many psychological and physical factors. The external genitalia of both sexes are very sensitive to touch, and they provide sensations of pleasure that are important to the sexual act. When the female is sexually aroused, the walls of her vagina secrete a fluid that lubricates the canal, making entry of the penis easier. When the male is sexually aroused, his penis becomes erect. Early in his arousal, a small droplet of fluid secreted by one of his glands cleans the way for the sperm. This drop may actually contain good sperm. If just that drop gets into the woman's reproductive system, she may become pregnant. The penis is inserted into the vagina. This is sexual intercourse. The partners move their bodies so that their sex organs rub against each other, heightening the sensations of pleasure and sexual excitement. Ultimately, each partner may reach a sexual climax called orgasm. For reproductive purposes, the climax of the male is particularly important. It is then that muscular contractions force a sudden discharge, or ejaculation, of semen into the vagina. Well over 100 million sperm cells are normally ejaculated at once. Each of them contains half the genetic information required to make a human being. Once inside the vagina, the sperm are moved along by muscular contractions of the woman's reproductive organs and under their own power. Their tails propel them forward. The rest of the semen serves as a nutrient to give the sperm energy. Of the millions of sperm that are ejaculated, only about 100 will make it as far as the ovum. Some will be killed along the way by the natural defenses of the woman's body. Some will run out of power. Some will be functionally defective. It is not fully understood how they are guided, yet many of the sperm move up into the uterus and then into the fallopian tubes. The sperm may live for about 48 hours, so that if an ovum is already in one of the fallopian tubes, or will be within 2 days, fertilization may take place.
The development of each new generation begins within a woman's body. It is here that two living cells, the egg and sperm, will unite to form one cell, the zygote. The fertilized egg, carrying a unique complement of genes from the father's sperm and the mother's egg, then commences a program of development which will convert this one cell into a complex organism. Within 9 months, the egg will divide repeatedly, forming a large number of cells which will follow precise and sequential patterns of movement and differentiation, resulting in the formation of a human being. This process of development begins within the pelvic region, where the reproductive organs are located. A closer look reveals the ovary, where egg maturation occurs, and the uterus with its outstretched fallopian tube or oviduct. The flared end of each fallopian tube, the ampulla, lies close to the ovary. Each month during a woman's reproductive years, egg maturation occurs in the ovary. In this organ lie thousands of tiny follicle units, each containing an egg cell known as a primary oocyte, surrounded by follicle cells. These oocytes have ceased to divide by mitosis and have been arrested at the metaphase stage of the first meiotic division. This transition from mitotic to meiotic division of egg cells occurs in a woman's ovary before her birth, so that these primary oocytes have been quiescent at least 12 or 13 years at the onset of puberty. After puberty, a few follicles mature each month until the onset of menopause, some 40 to 45 years later. While the development of several follicles is initiated each month, usually all die except one by the time of ovulation. Assuming an average menstrual cycle of 28 days, counting from the first day of menstruation, egg maturation takes place during the first 14 days of the cycle. The various stages in the entire process will be shown on the ovary in the drawing. In stage A and B, lasting 7 days, the primary oocyte enlarges and the surrounding follicle cells begin dividing. Gradually, on day 8, a fluid-filled cavity, the antrum, forms within the follicle layer of cells, as shown in C. The enlarging follicle approaches the ovarian surface as seen in D, and during the final maturation process, it bulges from the side of the organ. At stage E, the ripe follicle is shown on day 14, just before it ruptures to expel the egg. This oocyte has just completed the first meiotic division. The follicle and the egg here are drawn very much enlarged to illustrate developmental events. In reality, the diameter of the follicle is approximately 1 centimeter, and the egg is only 1/10 of a millimeter in diameter. The follicle surrounding the oocyte contains a large fluid-filled antrum. A combination of hydrostatic pressure from this fluid and enzymes from the follicle cells which weaken the ovarian wall result in rupture of the follicle and release of the secondary oocyte. This release is called ovulation. The secondary oocyte released from the ovary is drawn into the ampulla of the fallopian tube by currents created by the beating of thousands of hair-like cilia. Following expulsion of the egg from the ovary, the follicle again closes and seals. The follicle then becomes known as the corpus luteum and secretes steroid hormones, estrogen and progesterone. These hormones signal the uterine lining to thicken in preparation for the entry of an embryo into the uterus. The process of ovulation will now be repeated to emphasize important events. The egg will be slowly propelled by cilia all along the tube towards the uterus, a journey which will require approximately 5 days. However, the egg will survive an average of only 24 hours unless it is fertilized. If fertilization is to be successful, sperm must reach the upper end of the fallopian tube. Sperm are propelled through the reproductive tract by the thrust of their own motile tails, and in the fallopian tubes by peristaltic contraction of the tube walls. Of the millions of sperm released into the vagina, only a few hundred will reach the upper end of the fallopian tube. When they encounter the egg, they stick to the outer surface. Once the membrane of a sperm head contacts the egg cell membrane, the sperm is drawn into the egg, and all further penetration by other sperm is prevented. The first division occurs 18 to 24 hours after fertilization, as the zygote slowly progresses along the interior of the fallopian tube. As with each subsequent division, the number of cells doubles. Because the embryo does not increase in size with each division, the resulting cells are small. The solid ball of cells produced, known as a morula, begins to hollow out. The cavity forms only in the center of the cell ball, but it is shown here as if the outer cells were transparent. The hollow sphere is now called a blastocyst. A mass of cells accumulates at one edge. This pile of cells forms the inner cell mass, and it is from these cells that the human body will form. The remainder of the sphere, the trophoblast, will form the embryonic component of the placenta. "4 days have passed now since the egg was fertilized, and the blastocyst is approaching the uterus. In this drawing, the size of the egg and the diameter of the fallopian tube is enlarged for clarity. On the 5th day after fertilization, the blastocyst enters the uterine cavity. As a result of this growth and the erosion of the uterine tissue, the blastocyst penetrates the uterine lining. This process is known as implantation. By using a higher magnification to watch this process, the details of the enlarging blastocyst can be seen. This view shows how the cells have arranged themselves. Keep in mind the massive tissues you will see develop consist of accumulations of many cells, although they will not be shown. Within less than 1 week, the embryo buries itself completely within the uterine wall. The outer trophoblast develops into a syncytium, where many nuclei are found within the cytoplasm of the large cells. This is the syncytotrophoblast. The inner layer of the trophoblast, the cytotrophoblast, directly surrounds the inner cell mass and the trophoblastic cavity. The implanted embryo is now completely surrounded by uterine lining. The erosion caused by the invading syncytiotrophoblaст has ruptured tiny blood vessels in the area of the embryo so that it is surrounded by pools of maternal blood. A cavity appears within the inner cell mass and gradually enlarges. This is the amniotic cavity. A portion adjacent to the trophoblast is the amnion, and the thicker interior region is known as the embryonic disc. These cells of the embryonic disc eventually will form the body of the human embryo. These cells separate into three distinct layers: the ectoderm, mesoderm, and endoderm. This separation of the three primary germ layers is called gastrulation. By the 10th day after fertilization, the cells forming the endoderm begin to appear as a distinct layer at the periphery of the inner cell mass. This migration of cells from within the embryonic disc and their aggregation to form a separate endoderm is known as delamination. Delamination of the endoderm is the first part of gastrulation in humans. The layer of endoderm continues to grow and gradually forms a fluid-filled sac, the yolk sac. The name of this structure is misleading because the sac contains no yolk and does not function in the nutrition of the embryo. After delamination of endoderm cells from the germinal disc, the remaining layer becomes known as the epiblast. The epiblast contains cells which will become the ectoderm and mesoderm. The separation of these two layers is the second part of gastrulation and occurs by primitive streak formation. Cells of the epiblast will migrate toward the posterior midline to form a long pile called the primitive streak. They will begin to drop below the epiblast and accumulate between the epiblast and endoderm. The cells which pass through the streak will form the red mesoderm. Those which will remain on the surface will be the ectoderm. The growing mesoderm has split as it expanded. One layer has migrated over the surface of the yolk sac, while the other surrounds the amnion. Mesoderm adjacent to the endoderm is splanchnic mesoderm and is destined to become highly vascularized. The combination of endoderm and splanchnic mesoderm yields a tissue known as splanchnopleur. Mesoderm which surrounds the amnion is called somatic mesoderm. Ectoderm and adjacent somatic mesoderm together form somatopleur. The somatic mesoderm now lines the inner surface of the cytotrophoblast. At this stage, the embryonic structure consists of four parts: the trophoblast, which will form the extraembryonic placenta, the yolk sac and extraembryonic splanchnopleur, where both the blood cells and the germ cells will originate, the amnion, an extraembryonic somatopleur surrounding a fluid-filled sac that will form a hydrostatic cushion during later development, and the trilaminar sheet of ectoderm, mesoderm, and endoderm, which will form the body of the embryo itself. After the movement of mesoderm through the primitive streak ceases, the structure disappears and gastrulation is complete. The three distinct germ layers are now formed, and in the region of the embryo proper lies a trilaminar sheet, ectoderm in blue, mesoderm in red, and endoderm in yellow. Along the midline, the mesoderm segregates as a long rod, the notochord. This will become an early embryonic supporting structure along the back. Flanking the notochord along each side, the mesoderm condenses to form a series of paired segments, the somites. These will form the major muscles lying parallel to the spine and the bones of the spine itself. Biochemical signals from the notochord and the somites induce the overlying ectoderm to form the central nervous system. Along both sides of the dorsal midline, the region which will be the future back of the embryo, two ridges of ectoderm make their appearance. These are the neural ridges or folds. As they increase in size, the folds extend towards each other and make contact all along the back from the future head to the future rump of the embryo. Remember that you are viewing this process here in cross-section at a point about halfway down the back. Once the folds have made contact, they fuse, closing off a tube of ectoderm below the surface. This is the neural tube, and it will form the central nervous system. The surface ectoderm above the neural tube will form the skin of the embryo's back. Between the skin and the neural tube, an important group of ectoderm cells originate. They're too small and too many to show here correctly. These are the neural crest cells, which were located at the fusion point of the neural folds. They remain separate below the skin, but above the neural tube. They remain here only briefly, and then begin a rapid proliferation and extensive migration. Eventually, these cells will form such important and diverse tissues as the spinal ganglia and the peripheral nervous system, the adrenal medulla, pigment cells in the skin, and connective tissue in the head. To observe the embryo lengthwise, it will first be rotated to reveal the profile of the hemisphere. The uterine tissue and syncytiotrophoblasts have been removed, as we will concentrate on embryo development and identification. A sagittal cut along the midline separates the hemispheres in two. The sagittal view is then completed by filling in the remaining portion of the section. Much of the embryonic structure is spherical, so it looks quite similar in a cross-section or sagittal section. The embryo itself, however, is now seen lengthwise, with the anterior or head end to the left and the posterior or tail end to the right. The entire trilaminar sheet of ectoderm, mesoderm, and endoderm will begin to lift and fold to form a distinct embryonic body. Observe also that a posterior outpouching of splanchnopleur will gradually penetrate an area known as the body stalk. This outpouching is the allantois, another important extraembryonic membrane. While these internal changes are taking place, the trophoblast structure becomes more elaborate, forming a chorion or embryonic placenta. The extension of numerous villi provide a greater surface area for exchange of nutrients, gases, and wastes between the blood of the mother and the blood vessels of the embryo. As the embryo develops, the exchange requirement increases, and the surface area of the chorionic villi will be seen to increase to meet the need. As the embryo folds, the yolk sac is slowly pinched off, while the amnion expands to surround the embryo body. The connecting body stalk maintains the contact between the embryo and the surrounding trophoblast. The endoderm, which forms a blind tube in the head region, is the foregut. Its smaller counterpart in the posterior region is the hindgut. When the folding process nears completion, the connection between the yolk sac and the embryonic gut narrows to form the yolk stalk. The yolk sac now lies outside the embryo's body and therefore is clearly an extraembryonic membrane. The other extraembryonic membranes are the allantois, a splanchnopleur extending into the body stalk. The endodermal lining of the allantois remains insignificant in the body stalk, but the important allantois mesoderm has extended through the stalk into the chorion, providing blood vessels for circulatory exchange there. The amnion, also an extraembryonic membrane, consists of a somatopleur, providing a fluid-filled sac around the embryo, and the trophoblast, with its mesodermal lining which forms the embryonic placenta or chorion, where nutrient, gas, and waste exchange occurs with the mother. The lifting off of the embryo has brought the fold of the amnion into a position to surround the yolk stalk and body stalk with the enclosed allantois. This broad connecting structure between the embryo and the chorion is known as the umbilical cord. In the mesoderm beneath the foregut, formation begins with a hollowing out of two tubes, the heart primordia. At this stage, 4 weeks after fertilization, the body pattern of the human embryo can be clearly distinguished. The folding of the trilaminar sheet has resulted in a tube within a tube body plan, with an outer tube containing an inner tube of splanchnopleur. The inner tube, which will form the digestive system, is still continuous with the extraembryonic yolk sac and allantois. The openings of the future mouth and anus have not yet broken through. This view reminds you again of the position of the embryo within the uterine wall. Continued growth of the embryo will soon obliterate the lumen of the uterus entirely. The further development of the embryo will now be followed with the surface view of the body. The yolk sac is shown here without the surrounding splanchnic mesoderm, in order to distinguish it clearly from other mesodermal tissues. Here at 4.5 weeks, the limb buds of the arm and leg are visible. The presence of rubella virus or drugs such as thalidomide at this stage often arrests further limb development and have also severe internal consequences. Folds in the neck region are visceral arches, homologous to the gill bars of aquatic organisms. The otic placode is visible adjacent to the hindbrain, and the impression of the mesodermal somites is apparent under the skin along the back. The eyes will appear on the side of the head above the first visceral arch. The eyes will gradually be pulled forward to the front of the face by the movement of the visceral arches. At this stage, the embryo has a distinct tail structure, which will shorten and disappear before birth. As the limbs extend, flattened paddles form at the distal ends. By programmed cell death, the paddle is transformed into a hand or foot with 5 separate digits. The face is recognizable as a human's by 14 weeks of development. The actual size of the embryo is now 5 to 10 centimeters long. The position of the embryo in the uterus and the internal anatomy of the mother can now be seen at 24 weeks. During the remaining weeks of pregnancy, the growing fetus expands and stretches the uterus. The mother's abdomen enlarges to accommodate the space required by the developing infant. Cushioning of the baby's body is provided by the fluid encased in the amnion. As term approaches, the infant usually assumes a position in the uterus with the head adjacent to the birth canal or vagina. The film will now be reviewed again without narration. Try to mentally narrate the events yourself as they occur. Thus we have followed the reproduction of the human species. Complex physical growth and the movements of cells and tissues have been traced. The underlying biochemical explanations for these complicated developments remains largely unknown. The full understanding of the normal process of human development is the goal of considerable research worldwide. The eventual benefits of these studies will include prevention of birth defects caused by organisms, chemicals, or genetic influences, and contribute to our knowledge of how the body controls and regulates its components. When this regulation fails, as in the case of cancer and immune diseases, this knowledge is essential to successful medical intervention.